Starting us off for this term and indeed this year, we've got David Stansby from UCL, who's going to be talking to us a little bit about chunking in massive imaging datasets.
Cool. Thanks for the invitation. And yeah, hi everyone. It's good to be here. So I'm going to talk about chunk file formats for massive imaging datasets. So the image I put on the title slide, I think, is one of our most beautiful images in our project. It's a slice across the heart, and the whole heart image at 8 micron resolution. So if you zoom in, you can see individual cells. And this is the second largest data set. So it's a 3D image. Imagine it kind of coming out of the plane, and it's 8 terabytes in total. So I'm going to talk to you a bit today about how we try and handle and analyze these huge data sets. So I'm David Stansby. I looked on my website to find out who I am. I write open source software for research. I guess that's the kind of my big work identity. I'm worried about the climate. I'm a keen walker. That's my identity, or 2 parts of my identity out of work. And then in terms of projects, specifically, I currently run the Human Organ Atlas. So I'll talk a bit about that. So a brief history of myself in logos. I did an undergraduate here in Cambridge, so it's really nice to be back. I then did a PhD in astrophysics, more specifically solar physics. So I studied the magnetic field of the sun. And during my PhD, I got really annoyed at a bug in matplotlib and decided to fix it. And I kind of caught on to enjoying doing that. So matplotlib development is how I really got into open source software development, research, software engineering. It's where I learned a lot of the skills I use now. And I'm still on and off a matplotlib developer. So later over coffee, feel free to come and complain to me about what you hate. Lots of people enjoy doing that. I also developed a lot of domain-specific software. So SumPy I worked on is kind of the core Python library for doing solar physics. And so I adapt a big community effort. And then PFSSPy on the right was the first package I created for research software, and was for doing magnetic field modeling of the sun. And it's useful that the PFI paper is my most cited paper, so I'm preaching to the converted here with that, but proof that research software is valuable. I'm now at UCL, where I'm a senior data scientist technically, but I see myself as a software engineer. And in software, I work a lot on ZAR, which I'll talk about. That's a file format for storing arrays. And I run Human Organ Atlas. So this is the Organ Atlas logo. So today I'm going to talk a lot about 3D images. Just to orient yourself, it's hard to visualize 3D images on a 2D screen. So a lot of the time I'll just show you a 2D slice of the 3D image. So like imagine one of these great planes here. I'll just show you that. But imagine it's in 3D, and this is a volume rendering just so you can imagine that all the images I show in 2D are really in 3D. But what I talk about is applicable to 2D images, to 3D images, to 4, 5D. It's really quite general purpose. So Human Organ Atlas. So I thought I'd start by talking about what a clinical scan of someone's organs looks like. So if you go to a hospital and you have a scan, so that's a 3D X-ray essentially. This is what it looks like. These are my lungs. A prize if you can tell me what's wrong. This is kind of a volume rendering. And it looks really nice, but you can kind of, and the tube coming in the top is the main airway. But you can see the air inside the lungs. But you can see as you get down to smaller scales, it kind of gets pixelated. And you can't really track those airways down to kind of more than a couple of branchings. So every time the airway branches, it gets smaller. And you're limited by resolution. So mainly because you don't want to give humans too much radiation. CT scans are about 200 megabytes in size, say, so I can load this on my laptop, I can view it in 3D, I can do all sorts with it just on my laptop, which has 8 gigabytes of RAM. And the resolution of this is about a millimeter, so the pixels about a millisecond size. And if you just look at the 2D slice of that, this is what it looks like. So you can see stuff, but it's pixelated, right? If you're treating someone and the way you get information about human tissue is through a biopsy. So if you ever had a biopsy, you'll know that involves taking a small sample of tissue from you. And the advantage of that is you can look at that small bit of tissue in a microscope and learn a lot about the cells. So on the left here, you can see individual cells, and the scale bar is 50 microns. So a micron is 10 to the minus 6 meters. I'll use that a lot in this talk. So that's 1,000 times smaller than a millimeter. And you can see on the left here individual cells. And the other nice thing about a biopsy is you can access the physical tissue itself. So an X-ray is non-invasive. A biopsy is invasive, and because you have the tissue, you can stain it. So on the right here, you can see a biopsy that's been stained with a certain chemical, and it's bringing out a certain type of cancer cell. So this is one of the main clinical ways of diagnosing different cancers and other diseases. The trouble is, as I said, this is on a really small scale. So, like, the dream is to have this, but across a whole organ, right? And you can really then tell all the way from the level of individual cells, and then how those cells are organized into a whole organ, how that organ's working. So that's exactly what we do in the human organ atlas. So we do CT scans, so 3D X-rays of human organs. These are after people have passed away. So the radiation dose to do this is so high that you wouldn't be alive after it happened to you if you tried it. And we put all this data on the Human Organ Atlas. So to do this, you use really fancy X-rays, and we go to a synchrotron. So this is the European Synchrotron Radiation Facility in Grenoble in France. It's beautiful in the Valley of the Alps. And this ring here is the synchrotron. And essentially they fire electrons around that donut really fast that produce really good X-rays that allow us to scan organs, whole organs, down to the scale of individual cells. And so this is, you can't see super well because of the contrast, but these are some of our samples we were scanning a couple of weeks ago, one of our scientists. So we put our samples in jars, the X-rays come in, and we have some fancy cameras at the back there. So what can we do? So do this. So I'm going to try and get the science out of the way to motivate the software engineering later in the course. Two examples of stuff we've done. So in 2022, we looked at how lungs of people with COVID-19 were affected to try and understand the pathways through which COVID-19 causes death eventually. So you can see at the top right a control, so the lung of someone who died from something else that wasn't COVID. The bottom right, the lung of someone who did die from COVID. The scale bar is about 50 microns again, and you can see the airways in the lung on a very small scale. And you can see in the healthy lung on the top right, the airways are all very separated, whereas on the bottom right, they're kind of clumped together. And what we managed to show is that there was lots of kind of shortcutting ways, which meant oxygen wasn't being delivered as effectively in the COVID lung, which was one of the reasons why COVID caused such a high mortality. One of the other things we can do is really look at how these trees are organized across the whole organ down to tiny scales. So this is the kidney, and the arteries, and the colors you're seeing are a segmentation, a labeling of the arteries and veins. And you can segment these right across the whole kidney down to small scales. And one of the things we can do is look at, so there's kind of some biological jargon in the top right plot, but on the x-axis on the right-hand side is kind of the branching level. So on the right is the largest vessels, on the left is the smallest vessels, and you can see the number of vessels at each level, say. So you can see as you go from right to left, the number of vessels goes up. And this is the first time we've been able to produce a graph like this, all the way down, all the way from the one artery coming into the kidney down to the smallest scale at which those vessels exist. And we don't just image kidneys or hearts, we image all sorts of organs. So if you go on our website, I'll show you the website later, we have livers, lungs, prostates, spleens, testes, uterus, heart, colon, brain, kidney, uterus, and testes. So yeah, that's kind of a background and science motivation for why we're doing this work. So let me go back to that CT scan of my lung. So this is the blurry CT scan. If I now take some of our data from the Human Organ Atlas, but I've downsampled this to the same resolution. So if you went to a hospital and scanned this lung, this is what it would look like. When we scan it, this is what it looks like. So it's like, if you wear glasses and have bad eyesight, it's like putting your glasses on. I find it absolutely amazing. And you can see this is a lung that has fibrosis, and you can really see the intricate details and how they interact across the whole lung. So this is imaged at 20 microns. We can also take a smaller chunk of that, zoom in, and we can image that at even higher resolution, and that's what it looks like. So beautiful, beautiful images. So how big are these images? So this slice here is 7,600 by 7,600 pixels, and then we have a stack of 12,000 of these images that form our 3D image. So if you do the math, 676 billion voxels, pixels, voxel is a 3D pixel, and 1.35 terabytes for this data set. So yeah, the real question that I've been working on in a couple of years on this project is how do you work with such a big image when you can't load it all into the memory of your computer? So my laptop has 8 gigabytes of RAM. We have some very fancy desktops in our office that have 256 gigabytes of RAM. If you're really pushing it, you've got a lot of money, maybe you have a terabyte of RAM, but I can tell you most scientists don't have a terabyte of RAM. And you know, we don't just have one of these images, we have loads of these images. So this is the data in the Human Organ Atlas over time. So there's about 6 years on the x-axis, terabytes on the y-axis. And you can see in blue all the data we've taken, and in red all the data that's publicly available. So we hit 600 terabytes a couple of weeks ago, slightly scary. We're definitely going to hit a petabyte, and we've got enough funding to get there. And we also recently hit 100 terabytes of public data. So that's all on the internet for anyone to download and share. I always find this graph a bit daunting. My job is to turn the blue line into the red line and make it all publicly available. And we're getting there, we're getting there. And to prove that it isn't just one huge image we've got, each of these dots is an individual image, time on the x-axis, so over 5 years. And you can see we're routinely taking data sets that are over a terabyte in size. So yeah, all the stuff between those two lines on top is between a terabyte and 10 terabytes. So how do we store these images? Sorry, I'm just going to take a little break. Does anyone have any questions at this point? Yeah.
I was just about to ask what kind of compression, if any, do you use? But I can see that you're using PNGs. Well, at least on this slide, so maybe that kind of goes into...
we Okay, maybe I'll talk through this slide and then answer your question if that's okay. Biologists, right, you take a picture on your phone, it's a 2D image. And because everyone in the world uses 2D images, the technology for storing 2D images is like really fancy and advanced. So biologists like to store lots of 2D images. And so imagine this is your array, and each color is a different slice going down. And all you do is you save, like, say, a PNG file, call it 0.png. Then the next one you say 1, 2, 3, and you keep going. So you have a directory of like 2,000, 3,000 2D images, and that's how you store your image. And so this allows you to use like widely used compression that's out there. So we actually use JPEG 2000 compression to store our images initially. It's lossy compression. It was not my decision to do that. I'm going to distance myself from using lossy compression. Yeah, we use JPEG 2000 because it gives us really good compression ratios while still maintaining the image. But this is really how biologists like to store 3D images. So the problem with this is, let's say I want to read these 4 orange cubes, each cube here represents one value of an array. So let's say I want to read these 4 array values. I have to open 2 image files. And if those image files are small, that's kind of fine, right? But you end up opening that whole slice of data and throwing most of it away because you only want 4 values. And as you start scaling to larger and larger images, this becomes more of a problem is using compression, because decompressing the images is CPU intensive and can become a bottleneck. So our largest individual slices are 7 gigabytes each, and so loading one of those takes on the order of minutes. So this is actually a really inefficient way to access data, even if it works well for storing it. And so this says exactly what I've said. So every time you open a bit of data, you have to load the whole slice of data because the whole file is compressed as one. So if it's compressed, that's very slow. It's a lot of memory. So like every time you load a slice, you have to allocate memory for that entire slice, only to throw away most of that data once you've opened it. And our data is isotropic. So these 3D images are truly 3D. The resolution in each direction is exactly the same. And so it doesn't really make sense. We want to work with it the same along the z-axis as we do along the x and y-axis. So I've taken an example. This is the brain, the black circles. You can see blood vessels running through it. And if I wanted to load 128 cubed cube, so that's about 4 megabytes of it, I've got to load 128 slices, which is 20 gigabytes. So I load 20 gigabytes, throw away everything else just to get 4 megabytes. So I've laboured the point, but hopefully I've convinced you that storing slices, it doesn't scale well. The solution, and really the central take-home message of my talk, is the solution is instead to store your images in chunks. So instead of breaking up your array slice by slice and saving each of those slices as a single file, instead you break up your array into chunks, and these can have so here each color represents a different chunk, which is stored as an individual file, your file system, on your cloud storage, and each color is a different chunk. And so in this example, I've split up my array into chunks of 2 by 2 by 2, and each of those would be saved to an individual file. And this is what ZAR is. So the whole idea of ZAR, so ZAR is a storage specification for n-dimensional arrays. And really the idea is you take your array data, you split it up into these chunks, you can control the shape of those chunks, you compress each of those chunks individually, and then you save so individual. So you kind of get the best of both worlds. You still get decent-ish file compression, but when you load some data, you don't have to access entire slices of your array. And as a bit of a teaser for later, hopefully this flowchart is ringing some alarm bells. You think, oh, this is great because I can now read all these chunks in parallel. So it really lends itself to parallel computation as well. So using the same example of that brain, you only load chunks around the volume of interest that you're after, you have to load a little less data. If I have my chunks are 128 by 128, and I want to load a bit of data, the maximum number of chunks I've got to load is 8. And so I only end up loading 33 megabytes. And the other thing Zara has been optimized for is fast decompression and compression. So you can configure how your data is compressed. You can choose from a menu of compression and decompression routines, and they're all designed to be potentially slow to compress the data in the first place. So when you write the data, that's a one-time operation, hopefully, and it can be slow to compress them, but then reading them is always fast, no matter how long it takes you to do the compression. So these are designed really for write once, read lots of times, which is when you have open data is typically what happens. I'll put a bit of data on the server, and hopefully loads of scientists will come along and use my data and cite it. Okay, so that solves our kind of like, we want to access a little bit of this array problem. On top of that though, if we want to zoom out and look at this whole sample, we've still got a huge field of view, right? And so the resolution of my laptop screen is what, 2,500 by 1,300 or something like that. But our images are like, I don't know, 7,000 by 7,000. So there's no point loading 7,000 by 7,000 pixels is 2,000 by 1,000. And it's kind of like Google Maps, right? If you're looking at the world, there's no point loading the street over there because you'd have to load the street over there across the entire world, and it'd take forever, right? So the other key feature of these modern data is storing copies of the data at a resolution appropriate to what you're viewing it at. So you take the full resolution data, and you also store a copy that's binned by 2 or downsampled by a factor of 2. You store a copy that's downsampled by a factor of 4, and so on, and kind of as much as you want. And you might think this is a bit wasteful, and it is. But if you do the maths in 3D, you only end up storing a maximum 1/7 more than the data you started with. So it's a trade-off, but in terms of storage, but it's really worth it. And this extension to Zara, storing these multiple resolution data sets, which is called OME Zara. And so the idea is you use Zara to store individual arrays, and then there's a specification for organizing those arrays at multiple resolution levels. And so this is specifically for biology, OME Zara. OME is the open microscopy environment. So it's developed by biologists for biologists, and it really enables, as I'll show you, data visualization and processing at a huge scale. I know there's a lot of geophysical work going on in Cambridge, and there are similar efforts in other domains. So GeoZar is a kind of similar effort for geophysics. Okay, so I'm now going to attempt to do a live demo and see how good Jerome is. So wish me luck. So what I'm going to do is I'm going to go to our website and try and visualize one of our data sets. For you live. I haven't, okay, so just like a magician, I feel like I should prove there's no trickery. I'm not sure how to do that, but I haven't downloaded any of this data. I haven't installed any special software. This is just the Firefox web browser. Does anyone want to nominate an organ to prove I haven't downloaded the data already? Brain. I heard brain first. Sorry if you're right. Okay, right. So you can see all our organs, and then you can see all our donors. So I'll click on this one. I apologize for the slowness of our website. And you can see all our data sets. So you see a little thumbnail, and these are our overviews. So we scan the whole brain at 20, 25 microns, and then these are the where we zoom in. So I'll show you a whole one. Okay, so you can, on our dataset page, you see all the metadata, and you can click on view visualization. Okay, so this is one of our brain datasets. So as I said before, we're currently loading one of the lower resolution copies of the dataset, and you can see it's already loaded, so the internet's good. And right, you can already see this, and there's a link at the top, and this is amazing for working scientists, right? Because you copy and paste that link, put it in an email, and they can just start looking at this data set. They don't need to know what Python is, what conda, and they don't need to install any special software. They can just do this for a web browser. And so you see three orthogonal slices there, and a 3D view. The 3D view doesn't look great because you can see the jar mainly. But yeah, I mean, you can zoom in and hopefully you'll see as it loads, you load that higher resolution data. There we go. So yeah, that's I still find this amazing. That's really the power of these chunked data sets at multiple resolutions. So I'll take another pause there for questions.
Yes, maybe it goes a little bit back. But when you talk about the voxel, what is the voxel? Imagine it's a grayscale image, but yeah. How big?
And so each voxel in our data sets represents typically 20 microns by 20 microns by 20.
No, sorry, I meant what is the data, right?
So the data, 16-bit, unsigned 16-bit is how we store it. Yeah, a grayscale, yeah.
There's a question online from Sharon. I don't know if you want to just unmute and ask. If not, the question was, do you have to consider storing and sharing data in your work?
I'm not quite sure. What do you mean by storing and sharing data? Yes, yes, we do have to consider it. But do you have a more specific question? Yeah. While we're waiting, whether
privacy aspects of that.
Yeah. We in terms of the fact these are human samples, so all of our donors have consented to their data being used for research after death. All the data is fully anonymized, so that removes a lot of the legal burdens we would otherwise have with dealing with this data. And all of our donors so far are actually from Europe. So in the UK, data law is a bit different, and after your death, your families, your immediate next of kin can request your data is removed. But in Europe, we're allowed to, once the donor themselves has consented, we're allowed to keep that data public forever.
There was another question from Mike, which was, what tools do you use to create the website viewer with a Python backend? Is it Django?
We have a website person, and I am not the website person. Let me get back to you afterwards with a more detailed answer, because I can see our website source code and check for you.
When you chunk your data and when you use the lossless compression,
how do you make sure that you don't have an artifact in the buffer or chunks?
So if you use lossless compression,
Sorry, if you use
lossy, yes.
Not about
Yeah. So when we chunk the data, we switch to lossless compression to avoid that problem. If I'm not sure there are any compression, I'm not sure you can do lossy compression with ZAR, but that would be an issue, certainly. Yeah, I don't know is the answer, but we don't do that. Yeah.
That's when you chunk the data, you have a lot more files compared to if you have slices.
Yes, that's a really good question.
If you were to still want to do like a very long
Yeah.
read across the whole cross-section,
Yeah.
wouldn't that be longer than?
Yes, so if opening all those files took a lot of time, then yes, it could be longer. Especially if you're using remote data, that can be a consideration. I'm not going to talk about it here, but when I talk about chunks, that's how it worked in ZAR version 2. There's a new version of ZAR version 3 where you can put more than one chunk in a single file. And I won't go into more details than that now, but we could chat afterwards if you want. But you can, ZAR v3 will keep the chunks but have less files.
Would it be possible to convert those to some polygonal meshes, like tables?
Yeah, so you can, it's just array data, so anything you can do with other array.
Would it be useful?
Yeah, possibly. Yeah, we haven't tried converting the data directly to meshes, but I imagine it would help, yeah. Because you could, yeah, I imagine you could mesh each chunk individually and then stitch the mesh together. I'm going to stop thinking on the fly because there's probably someone who knows more than I do about meshes. Any other questions? Any more online? No?
Sorry, one question.
Yeah.
In ZAR, do you have a possibility that in some way you have a different regions and different resolutions in the one image?
Not in ZAR version 2. ZAR version 3 is a lot more extensible. So you can how your chunks are stored in, how your chunks map to files on the files. So it might, but I've never tried it. And it would be complicated. But it might be, yeah, it might be possible. Cool, I'm going to carry on then. Okay, so that was a demo. You can go to the Human Organizers website and do this on your phone or your laptop yourself if you want and look at any of our datasets. Cool. So back to the talk. Okay, and you can annotate these datasets to label them. I'll skip over that in the interest of time. So the final bit I want to talk about is how this data storage format lends itself to processing data. So this is with thanks to John Sporing, who's at the University of Copenhagen. We have been thinking about this together for a few months. So because your data is split into chunks, it's kind of ideally suited to parallel processing. And so if you think of converting one image and processing somehow and having an output image, the key things are mapping from the chunks in the input image to the chunks in the output image. So there are a couple of different families of operations you can do. So the first one is like a point operation. It must have it, but it's where you directly map from one array element to another array element. So an example could be, if I want to divide my image by 2, I just loop through every array element divided by 2, assign it to the output array. And so each chunk, so chunk I, I'll do it in 2D just for simplicity here. Chunk with an index ij just maps to chunk with an index ij in the output. And you can basically just parallelize this over every chunk in your image. If you want to downsample your image, so if you want to take basically every square of 4 pixels and create a bigger pixel to create those downsample data sets, you basically are mapping from 4 chunks in your input image to 1 chunk in your output image. And again, this is kind of like a quantum, a unit of work that you can apply across your whole array. And you only need to load in the chunks in this graph. You're loading those chunks at any one time, which limits the memory usage. And if you want to upsample, you are doing the opposite. So you're taking 1 pixel and you're basically just copying that value to 4 others. And an area where this is useful is you might want to do an operation on a lower resolution version of a data set, just to make it quicker, and then you want to apply the result to a higher resolution. So masking out, say, the organ is one operation where we do this. Finally, convolving. So I haven't drawn the flowchart because I couldn't be bothered drawing out 10 input chunks. But you take a chunk and you need all the adjacent chunks. So imagine you're taking a filter and scatter it. When the filter's at the corner of the chunk, you also need the values from its neighbors to compute the result. So essentially you need, in 2D, you need 9 chunks to create 1 chunk. So you can kind of come up with a guess as how much memory this is going to use. So in 2D, for each operation, I've listed the number of chunks. So you need to read 1 and write 1, or read 4 and write 1. And if you have 8 megabyte chunks, you can kind of guess how much memory you're going to use, right? And so when you have these 2 terabyte data sets, this is really useful because you can say, oh, I've only got 8 gigabytes of RAM. How can I process this 2 terabyte data set? How many operations can I do in it at once? And so this is the idea we came up with. Like all good ideas, as I discovered during my PhD, someone's already come up with it. So I discovered a few weeks ago this library called Cubed. If you're a Python programmer and you have used or heard of Dask in the past, this is a drop-in replacement for Dask. And it has 2 unique selling points. It estimates the memory you're going to use ahead of time. So you set up an operation and it says, I think you're going to use this much memory. And then it will either go, oh, you don't have enough memory, rethink what you're doing, or it'll be like, great, it'll probably work. And the other thing it does well is it is really designed to scale well with a number of jobs. So if you've ever tried to use Dask and you have like 1 million jobs, it goes horribly wrong because it tries to construct this huge complicated graph with 1 million nodes, and it just doesn't like it. Whereas Cubes has kind of been designed to scale really well. The drawback is it's much less general purpose than Dask, but for our purposes, for image processing purposes, it works really well. There's some good intro slides on their website. I have nothing to do with this library. They came up with this before we came up with the idea, but yeah, I would encourage you to have a look at that. And so, yeah, that's kind of almost the end of my talk. So one of the other things I've been doing, apart from using these file formats, is working out how to make it easier for others to use them. So I had some EU funding recently for a project to teach these right resources and tools for working with these data sets. It's called Hefty. This acronym is the pinnacle of my scientific career because each letter is the first letter in it, so it's a good acronym. And it stands for Handling Enormous Files from 3D Imaging Experiments. And there's an elephant, because elephants are big, and some cubes, because of the chunks. So really the big thing we did was write a little, what I'm calling a digital textbook, which I'll show you quickly. and it kind of goes much more into depth in all the topics I've touched on in this talk. So here it is. It talks about formats, how to create the data sets, how to do parallel processing with them, and then exporting them to file formats. So if you have tools which don't understand these data sets, you can do some processing and then export them. So yeah, it's kind of a Python notebook, so you can run the data, run the code, sorry. Yeah, I'll post a link in the Zoom chat once I finish talking. Cool, so I'm going to stop there. Thanks very much for listening. I'm super up for questions and chatting, and thanks for inviting me.
Thank you very much, David. I realise we answered these, but if anyone has any more, please do stick a hand up, or if you're online, stick a hand up and you can unmute and ask, or drop something in the chat.
It's not really a question for the speaker, but I'm just curious as to maybe Chris or Simon Clifford's online. How related is this to like the biodat imaging project stuff? Like the problems that are being.
I think.
It looked very similar from my very cursory understanding.
Is Simon online?
There is a Simon. I don't know whether it's Simon Clifford or Simon Lee. Yeah, there is a Simon, but I'm sure you can talk to Simon afterwards as well.
I'm curious to know what.
Yeah.
I don't think Layla is online.
No, she can never make it.
Yeah.
Yeah, super happy to chat with anyone who you
It
think might
sounds
be
very
interested.
similar to the problems that they had to deal with as well.
Yeah.
So, pursuing that line of questioning in everybody else's absence, the point, the library that's not yours, the
Cubed.
Cube, that's one.
This one.
Yeah, those operations.
Yeah.
You were talking about upscaling, upsampling, downsampling. Could you do more complex? So can you compose operations into sort of more complex kernels?
Yeah, you can, and it's lazy evaluation as well. So you basically can write your pipeline. It will create an execution graph. It will estimate how much memory that's going to use, and then you can execute that either on your computer or on a cloud service.
Can you do it in multiple nodes on the cluster? So say one node only had enough memory to it.
Elena is online and said Simon Clifford used Dask and Zar.
Okay, right.
Yeah. Yeah, I think certainly Dask you can, like there are executors for like Slurm clusters, but I'm not sure about Cubed.
Got one saying, have you explored SciPP, which is based on Xarray, and compared that to Zar? Xarray, I presume, using Dask.
Yeah, I have never heard of SciPP, but I will check it out. Thank you for the recommendation. Yes, have not.
It's a bit of a rubbish question, but do you have a favourite image?
I have a favourite image.
Favourite organ?
That's a really good question. I really like the heart. The hearts look really cool.
In your app?
Favourite image? Yeah, I'd say the hearts. The hearts look cool. They have you can zoom into the muscle and see the individual cells that cause the heart to beat, which I think is really amazing. That was a good question. That wasn't a rubbish question.
It's a bit less technical, you know.
Super. In which case, you mentioned the usefulness of these tools to work with 3D biological images. Do you know if people are using them for processing scientific videos?
Ooh, I do not. No. I imagine it would be useful for videos. If you have huge datasets. I mean, a video is just a 3D array where one of your axes is time. So you might want to, because you might want to treat your time dimension differently to your spatial dimensions. You might want to like have a different, maybe your chunks will be a different size along the time axis or the XY, but yeah, you definitely could.